hope you've had a good week.
Yeah, it's been great, thank you. Busy, but good.
Yeah, I know that feeling. Beginning of the week, I was going to do all sorts of things, write papers, write grants. No idea what I've actually done. But anyway, we're here today, and we're going to talk about DNA sequencing.
We are, yeah, absolutely. And so I think perhaps before we dive into the details of DNA sequencing itself, perhaps for those listeners who are not experts, maybe we should perhaps discuss what DNA is and why we might want to sequence it.
Well, we've got a couple of real experts coming in later, which is going to be exciting. We'll introduce them when they turn up, where we're going to talk to them. But actually, you know more about DNA than me, so I can play the idiot here. Not
play.
So DNA obviously is the kind of molecule that codes for life. It's a self-assembly instruction booklet for an organism, isn't that right?
Yeah, absolutely.
And what I know is it's like a ladder that's been twisted. So it's a famous double helix spiral.
Yeah, sure.
And so what are the up bits of the ladder and what are the cross bits?
Okay, so the up bits of the ladder is what we call the backbone. And that's made up of two different types of molecules, a phosphate and a sugar, and they don't change, right? So the up bits are always the same. However, the rungs of the ladder are what we call bases. And those bases do vary. There's 4 different types.
Yeah.
And it's that sequence in which they appear that where we get this idea of the code.
And so amino acids are the building blocks of proteins.
Yeah.
And these are all molecules.
Yeah, okay. Yeah, so you're right, absolutely right. So we have these 4 bases, and the sequence in which they appear carries a code. The code is the blueprint for making these 20 different amino acids. And you can think of the 20 amino acids as 20 Lego bricks with which we build proteins. And these are molecules that we sometimes refer to them as the workhorse of the cell or the body. So they carry out very many of the functions and make us look like we do, and all the things the body does. A lot of it is done by proteins.
And so how does the code work? So how do you go from some bases, a string of bases, so they're called, I'm going to get this wrong, adenine,
Yep.
cytosine,
Yep.
guanine,
Yep.
thymine?
Yes, excellent.
Oh, yeah. Okay. And I struggle to remember those. And those 4 somehow code for 20 amino acids. So how do we go from the 4 types of base pairs, adenine, cytosine, guanine, thymine, so that's ACGT,
Yes.
to an amino acid? Not the whole kind of complicated molecular mechanism, but what the code is.
Sure. So the 3 of those bases will gives us 1 amino acid. So it's the blueprint for 1 amino acid. So the different order in which they appear.
Yeah, okay.
And so there's machinery in the body which will, again, without going into the details, which will read that order and know, oh, I've got these 3, so that means I must make this amino acid. And so it makes it, and then there's something else which adds them all, connects them all up, and then makes the protein.
Okay. So you have a chain of DNA, a double helix of DNA,
Yes.
made of these bases, and then that's translated into a chain of amino acids.
Yeah, there's a bunch of processes, but basically that's what happens. Yeah. And these amino acids make proteins, and the differences in the proteins you have, for example, compared to the ones I have, and the different numbers of them make us different in the ways we are, the ways we differ. We look differently, we have different characteristics, and that's a function of this code and these proteins.
But we only use kind of one strand, because it's a double helix, but we only use one bit of that for reading the code. Is that right?
Yeah, at a time. So you split them off,
yeah,
and then it gets read. And actually, the really interesting thing is that the strands come from our parents, yeah. And so this is why siblings will look alike, but actually we don't look. We're not all identical because there's differences within that. But it's we get once, the strands come from our mother and our father, and they get combined.
And they get mixed up, don't they? So
yes.
Even though we've got half our DNA from our mum and half our DNA from our dad, it's not like we've got one half of the helix from mum and one half isn't. No. I mean, because I used to think that.
Yeah, me too.
Yeah. I think that's a common misconception.
Yes.
But so and when an egg or a sperm is made, they're a mix of. So we've got two chromosomes each. We've got chromosome pairs, don't
That's
we? So I
right.
suppose we need to take a step back and mention that. So that means we've got. So DNA is snipped up in the body into like chromosomes.
Yes.
And we've got 23 chromosome pairs. Is that
That's
right?
right. Yeah.
Yeah. Okay. And one chromosome in each pair came from our mum and one from our dad.
Yeah.
But my mum's chromosome, the one that came from my mum, would have been a mix of her mum and her dad. So it would have been my grandparents. Is that right?
Yeah, absolutely. And so it gives us actually quite a repertoire of different characteristics that can come from all those mixtures. But of course, because they come from our parents, excuse me, siblings will look alike.
Yeah. My sister got all the looks and all the brains.
Mine got the normal coloured hair. That's not quite how genetics works.
So it's an amazing molecule.
It is.
It's also, it's very stable, isn't it? It doesn't react with. I mean, it's kind of very good at keeping the code, but it's rubbish in getting involved in chemical reactions.
Yeah, I mean, the code is protected, right? You know, it has a certain shape and morphology, we call this, and it's all coiled up and tangled up, so it's not really able to get, as you said, involved in other things that happen in the cell. The bases are very well protected. And one interesting fact about DNA is that the DNA we have in a single cell, if we were to uncoil it all up and unwind it, actually is 6 feet long.
Oh, wow.
In just a single cell.
No, that's amazing. And how it's billions of base pairs, right?
It is, yeah.
Billions of bases in each of our DNA.
It is, yeah, and that varies with organisms. So because we're complicated, highly evolved organisms, we have many of them. Simpler organisms like bacteria, for example, will have shorter DNAs and fewer bases.
Yeah, okay. And then you've already said this, we share, if we had an identical twin.
Yes.
I don't, do you?
No.
No, but they would have the same genetic sequence as us, an identical twin. Our siblings have about half the amount of DNA. We share 50%, half of our DNA with them.
Yes.
Ditto with our parents.
Yes.
And then 25% on average with our grandparents, but it can actually vary.
It varies,
it can vary.
Yeah.
But we can use that because we can build family trees by looking at so some bits of all of our DNA is identical, right? There's conserved genes, which if they break, then we don't function properly.
That's right.
Yeah,
yes.
Okay. But for the variable bits, those, when we're talking about sharing 50% or when we look to see how much we share with other organisms, they're the bits that vary between organisms, right? Yeah, okay.
And we can use those variations to detect, for example, origins, so where we deviated from each other, for example, where there are similarities. One can build up historic, almost, if you like, family trees and this sort of thing, just looking at DNA. It's actually odd how much the DNA is shared between organisms and entities that are completely different in appearance, and so it's fascinating, I think, and actually that also makes life difficult, because if we want to use the DNA to understand differences, so much of it is the same.
Yeah, okay. Right? No, I see. Yeah, but what scientists have done, and this is kind of closer to things that I've been involved with DNA, is you can work out not just family trees between individuals, but family trees between species.
Yes.
And in fact, when they've done that, they've estimated that the genetic code that we use today, that all life uses today, evolved 3.9 billion years ago.
Yes.
And I think one of the things that's really interesting about that is Earth kind of only appeared formed about 4.6 million years ago. So it didn't take that long on the grand scheme of things for life to get going.
That's amazing.
Yeah.
If we think about the grand scheme of, as you said, the life history of the Earth.
Yeah. But one of the big challenges is we'll talk more about sequencing with our guests now. But I think after that, one of the things is trying to understand how the genetic code got started and how life got started. Because as you've just explained it, the chemistry is incredibly complicated, DNA replication and what have you. And it certainly didn't spring into existence from nowhere. So we'll talk to people who who have been thinking about how life got started and how
Yes.
genetic code. But let's just talk about sequencing, because we're going to talk to some experts about that. That the sequence is the string of the bases, isn't it? It's the A, C, G.
It's the order.
T's,
It's the order in which
Yeah.
they appear, and that, it's just like it's almost a little bit like a computer, really, because it carries a code that can be interpreted
Yeah.
or a way of holding any kind of information.
Or even kind of letters in a book, I said. But there's
Yeah.
only 4 of them, and there's no spaces.
That's right. It's a very limited language in a way, but
Yeah.
actually because they encode for amino acids, then that language
Yeah.
becomes vast.
But not all of our DNA makes amino acids, does it? There are
No.
other bits and bobs in there that do other things.
Absolutely. It's a tiny, tiny proportion. The vast majority is non-coding.
Oh, I didn't know. Okay, I didn't know. Yeah. But some of that instructs. It's not useless, that DNA.
Yeah,
So-
it does other things like starts off other processes, creates a template for RNA, for example, and RNA, which is a molecule that carries out other functions that help with. It could help with DNA replication. It could help with protein synthesis. Lots of other functions that happen, but it doesn't encode, as you said, for amino acids.
But because in our bodies we've got like lots of different cell types. They're all come from the same. Each cell has the same genome essentially,
That's right.
but we've got like 220 different cell types. So in each one of our cells, and we've got like 37 trillion cells in our bodies.
That's right.
In each one of those, there's 6 foot of DNA,
Yeah.
and a fraction of that DNA creates proteins, amino acids that form proteins. Not all of those cells have the same proteins or use the same proteins.
No, not at all.
So how, in some cells, do we make some proteins, and other cells our DNA makes others?
Yeah, you're absolutely right. We've got vast quantities of DNA in every cell, right? But then this non-coding part of the DNA has these other functions. So, for example, has regions that proteins come along and bind to, and they can enhance or accelerate or initiate or block the production of these of amino acids. So you can think of it a little bit like a factory. All this stuff inside our cells is going on all the time. And not all of it is to make stuff. Sometimes it's to stop things being made. And this is how things are regulated with these other parts of the DNA that don't make or code for amino acids. They have these other functions.
So what's going on in our cells is way more complicated than what goes on in, say, my iPad.
Vastly so. And I think one of the mind-blowing things about biology when I first started to sort of delve into it and understand is, if I'm sitting here now talking to you, it looks like I'm static. But the sheer amount of movement that's going on at a very small level inside all of my cells is vast. There's things moving around all the time. The DNA is being replicated, the DNA is being read, and amino acids are being made. They're all being connected up, proteins are being made, and the whole thing is somehow regulated and choreographed.
No, it really is amazing. And I think what's incredible was when I was at school, and when Rich, our wonderful producer, was at school, we got taught Mendel. They knew what DNA was. They knew it was...
Are you sure they knew what it was in those days?
Thank you. Yeah, Darwin had just set off on his boat. Yeah. So anyway, Mendel had... we were taught Mendel, and we knew the structure of DNA, but everyone thought there was probably just a gene for everything.
Yes.
Right? So there would be a gene for how tall you were going to be, a gene for how intelligent you are. But what we've learned now is actually it's incredibly complicated. And we've learned that through a mix of DNA sequencing and a mix of looking at the products that DNA makes. And so one of the exciting things is we're now going to talk to some experts.
Right,
Okay,
Q,
let's
they've
go
just
let them
arrived. Okay, so Tim, now we have 2 real DNA scientists here with us, experts, in fact, world-leading experts. And shall we let them introduce themselves? So over to you, Gordon.
Thank you very much. My name's Gordon Sanghera. I am one of the founders of Oxford Nanopore Technologies, which was a spin-out from the University of Oxford 20 years ago this year.
20 years, wow.
Hi, thank you for having us. And my name is Lakmal Jayasinghe. I'm the CSO of Oxford Nanopore. I was one of the very first scientists to join Oxford Nanopore, a first wet lab scientist. And I've been there for 18 years as well.
I've just come from a government meeting, a lot of acronyms. Chief Scientific Officer, CSO.
Thank you, Gordon.
I shall be bringing him back down out of acronyms.
Well, this is wonderful. I mean, I'm quite, sort of taken aback you saying 20 years, because I think we first met each other maybe about 18 years ago when things had started moving. And on that vein, DNA sequencing is something that's been around for a long time, right? So can you tell us a little bit about why next generation sequencing is needed? This is what you do and what your company does.
Absolutely. And I'm going to trivialize the science. And it took 10 years and it cost several £billion to make this happen. But the way that you read DNA, you read it 20 years ago, and a lot of it's still done the same today, is you take something like a human being. We have 2 billion letters, GTAC, that's your source code. And you shrink it down, chop it up into 200 letters on a string. In reality, there's 3 billion in that one string. And then you use color chemistry, optical systems, large complex laboratories, lots of scientists, multi-million-dollar infrastructure and million-dollar equipment to decode and piece those 200 letter-sized pieces back into your 3 billion letter puzzle. What we do is actually read the DNA in its native form. What do I mean by that? We'd still chop it up, but we chop it up into very long pieces, like tens of thousands or hundreds of thousands of bases, and we don't do anything else to it. And we use a method called nanopore sequencing, which I'm sure Lakmal will articulate later, which allows us to read native DNA in its natural form, the way it exists inside an individual, rather than chopping it up and maybe getting a crude black and white approximation. You're seeing your DNA biology in high definition and full color.
So the traditional way would be like if you had all the separate words for a book, trying to put the book together. Is that all?
Indeed. And I think you could take it one step further. You've almost got seascape and skyscape.
Oh, yeah.
And it's all almost the same blue.
Yeah.
So you're putting the sky where the sea should be and the sea where the sky should be. And that exists today in existing recoding of the biology when you smash it up and then reassemble it.
So what's the longest read you can do at them? The longest string, say hundreds of thousands now, is it?
That's right. So the world record is 4.5 million.
Oh, wow.
DNA bases. Remember, it's 3 billion on one chain.
Yeah.
And the technology would and could eat all of that. Think about somebody sucking spaghetti. This machine can suck a 3.5 billion lettered spaghetti piece. But the problem is it's very fragile. So just the action of taking it from your body, extracting the human DNA, cleaning it up, even pipetting it onto the device, breaks it up.
Right.
So that's why tens of thousands or 100,000 bases is straightforward. But there is an ambition to read ever-increasing lengths. There's lots of interesting biology you get from reading intact genomes.
So Gordon, if we take things one step further, so Tim and I have discussed DNA and the genetic information that it holds, and you're saying that you've developed these techniques to read the DNA and sequencing it. Can you tell us a little bit about why do we want to sequence it? Why do we want to know what the code is?
So if you think about DNA and RNA, and just to give you a distinction, DNA is the source code. So that's your complete music catalogue, your code. RNA is your current music playlist. So you want to know both. And all diseases, our predisposition to diseases, our efficacy of treatments, even developing new drugs, and whether we're going to get these aging diseases like neurodegeneration, They're all hidden and wrapped up in our genomes. And today, with this short read black and white sequencing in legacy systems, about 25% of all human genomes, some of the toughest ones, pernicious cancers, neurodegeneration, and many, many others, are wrapped in what is referred to as the dark genome.
Okay.
I've not heard that expression.
That's right, it's a real thing. And because with short reads, you miss all these things. And so shedding light on those will have a huge impact. And we really are, even 20 years after the first human genome was mapped, still scratching the surface of our understanding of our source codes.
Sorry.
So you're saying that inside our DNA, in the code and in this dark genome, there's information that can perhaps tell us about which diseases we're predisposed to as part of our makeup, and which treatments would be more effective for us compared to other people. And that's a big motivation for understanding what's inside us, what's inside this code. Is that right?
That's right, 100%. But don't forget, our DNA is dynamic. Yeah. So over our lifetimes, we have GTAC. There's something called the fifth base, which is methylated, so it's chemically corrupted C, and that is a smoking gun in all cancers.
Oh wow!
And so
you can detect that in your screen.
That's right. You cannot do it with the existing systems, and you can pick that out, and that's a really exciting piece of biology that everybody's starting to think about and look at, and that modification is important. So DNA is not dynamic, and that's, if you smoke one cigarette, 4 of your C bases are corrupted. Now, don't worry, you've got 3.5 billion divided by 4, so there's quite a lot there. But it's the external factors, your lifestyle, your diet, all of these things can be part of that. And so it's not just what we are, how we were built from our parental hereditary genes, but what we do and how that becomes corrupted or not, depending on your lifestyle during the course of your lifetime.
To add to that, like when we talk about why DNA sequencing is required, so the human genetics is a big part, but beyond human genetics, what you can do with in agriculture and sequencing plants to see which soil condition is suitable for different crops and different varieties, and to monitor environmental samples to see what's in there, and to monitor water samples and sewage waste samples. And so applications are huge, but obviously human genetics and diseases is a big part of it.
So it's not just our sort of human DNA that we want to sequence, but that of plants and animals and other organisms too.
Can you give us an example? I mean, you, as well as producing the technology, you're also involved in some of the research that's doing this. Is that right? You're doing some work, I think, in Sri Lanka at the moment. Is that
right? So I think COVID is a good example. Like I think when it started in Wuhan, first thing they did in China was using MinION to characterize the COVID sequence.
And MinION is one of your devices.
Yes, so MinION is basically this one. So this is our latest version. So this is just handheld device.
Sorry, wait a sec. That does all this. That will read your billions of base pairs.
Exactly.
I thought you were going to be dragging in a wardrobe-sized device.
That's why we need this technology, because the other technologies have this huge fridge-size devices.
Can you hold that up to the camera there, just so you can see quite how?
Let me do this. Yeah. So that's our device.
It's small. It's smaller than a mobile phone.
It is. Yeah. This is our smallest version, portable version, and we have different scale devices as well.
Sorry, I interrupted you. You're telling us about
So during the COVID time, it's mainly like this that went around the world. So when it started in Wuhan, like obviously they went into using this, and at one point, like most of the Chinese CDC labs using this machine to characterize COVID, and then across the world, like more than 120 countries used this device to characterize DNA. So if you the the familiar words like alpha, beta, gamma, like Omicron, all the variants, and which variant is spreading in different countries. So to understand that, you need to sequence COVID virus to understand the mutations. And so we had like these end-to-end workflows. So we develop all these things so the customers can very easily sequence, COVID sequence, and understand the mutations and understand which variation is spreading in certain communities. So that was huge. So in Sri Lanka, actually, like when I got to know that South Asia entirely, like really struggling with sequencing capacity, so I contacted my friend who was leading this only lab in Sri Lanka capable of doing DNA sequencing. And she was by then using a different technology, one of our competitors, and they were spending $400 a sample. And as a result, they could only do 100 samples a month with the available funds. So they were really struggling. And then I talked to Gordon and we donated one of our Minna and Mark 1C packages. And then immediately they could do like 96 samples a day.
Wow.
That's a huge difference.
Cost came down from 400 samples to $400 per samples to $5 a sample. And more importantly, because it's very easy to use this machine, our sample preparation is quite short. They could go home. They said when with the previous technology, when they come early morning, they can't go home till midnight. So it was quite cumbersome.
So it was a complete game changer.
Complete game changer. And then the government got to know that there is this very easy method, and they using WHO funding, they set up 6 labs. In Sri Lanka, some of those labs were in very rural hospitals and kind of makeshift labs in a truck. And they didn't have any, where I went to see some of these labs and you couldn't do anything else other than putting one of those on this little space they had. So it was a complete game changer. And so within a couple of months, entire Sri Lanka was on Minna. And that was other, like especially third world or low and middle income countries. And 50% of all COVID sequencing was done in Africa, was done with nanopore sequencing.
Wow.
So that was quite something.
And so now that they have this capability and they can do this high throughput sequencing, this is going to help clearly with pandemic preparedness for the future.
Yeah, so that's a big thing now, right? Like pandemic preparedness for the future is in every country. They are trying to do something. So people who have this capacity, who have these devices already set up in their countries, in their labs, and they are using it for different things. But when the next pandemic hits, so the system is there for them to.
Yes.
But presumably not just pandemics. Presumably you can take this into any hospital and you can work out what illnesses, individuals that people are suffering from.
Yeah. Well, I was at Guy's and St Thomas's this morning. So before we touch on that, we sent 200 of these sequencers to Wuhan at the start of the pandemic. There's a very important point to make. The equivalent incumbent machine will cost you $250,000. These we sell for $5,000.
Wow.
So the important thing there is when you think about disruption and tech revolutions, this is the equivalent of moving from a mainframe computer, which is the preserve of rich nations and large multi-million-dollar international businesses, to a PC, affordable for everybody. And if we are going to prevent the next pandemic and the fight in the last pandemic, everybody needs to have access. So part of this revolution, because it is a tech revolution, is the social impact of affordable, accessible, democratization and access to DNA, RNA information. Back to London. So at Guy's and St Thomas's, what started out as a brilliant clinician, infectious disease clinician, Professor Jonathan Edgeworth, looking at long COVID patients, trying to understand what are the co-infections they had. That trial continued post-pandemic, and in the last 3 years, they have been using this device in ICU to look at respiratory infections. So they get a patient coming in with a respiratory infection and they send a sputum down to the lab in 4 to 6 hours versus 48 hours, 2 days. They return a result. It takes 3 days in the conventional method to say there are drug-resistant strains, antimicrobial-resistant strains of pathogen, so a broad-spectrum antibiotic will not work. They do all of that in that 4 to 6 hours. And then now we have precision infectious disease management, targeting the patient individually, and they demonstrated 45% of patients were on the wrong antibiotic.
45%.
45%.
Wow.
Now, but there's an additional bonus. If and when you see the next pandemic, you will see it emerging in intensive care, the most vulnerable. And it's almost certainly going to be a respiratory infection. And what you will see is this new variant that we haven't seen before, but it will be there, and it'll appear in Brighton, and it'll appear in Manchester, and it'll start popping up everywhere. And it's the same. And the difference here is, instead of doing what you do in conventional diagnostics, let's test for pneumonia, or let's test for E. coli, or this thing, it does a scan. It's called a metagenomic scan, and the net pulls in everything. And then you can see everything in there. And then you can say, okay, here's some drug-resistant strains, here's co-infections, multiple infections, here's something new and novel. So you start to get this additional benefit of clinical and biosecurity.
So when you 20 years ago, when you set up Oxford Nanopore, were these things you could see down the pipeline, or is this kind of, you could reinvent a bit of history now if you want, you said, yes, that's exactly what I wanted to do, or is it just, have some of these things come up?
I think so this is my second spin out. So I'd been involved with the company that helped transform type 1 diabetics testing at home, which is really important for blood glucose control and insulin injection. But the electronic methods and the scalability of that technology, the first thing I realized was we could apply that here. So we didn't think too much about applications. We thought about how can we take a brilliant academic experiment that is limited, complicated, in a laboratory with 4 PhD students running it, into a ubiquitous device that anybody could use. And that was the first 5 years. But we always knew, and we always had an eye on the fact that rapid insights in infectious disease, in critical care, near the patient would be something that would be huge. And so we always kind of knew that would come first, and it pretty much is our most advanced commercial application.
So,
Everyone wants to talk at once.
I know.
So taking it to a different level, like I think even NASA took this to International Space Station to sequence in space. So that was the first sequencing done in space to see what's out there, or even to monitor the health of astronauts. So
That's wonderful. And I think, sort of from what Gordon was saying and what you're saying, this idea of this being an academic experiment with 4 PhD students brings me sort of into the idea that I realise that we haven't actually touched upon what the technology is. So maybe, Lakmal, you can tell us how does it work? So we know it's a nanopore, but what does that mean?
So in the technology, so we have what we call flow cell, which is a chip. So in this chip, we have a membrane. So the membrane is quite stable. We kind of, rather than using, in body, you have like every cell has a natural lipid membrane, which is quite unstable. And that's what we use in all the academic experiments. If you go like closer to this, the complicated setups and bilayer breaks, is that unstable. So we had to make it very stable enough so that we can send around the world, ship these things to all the countries. So we build the membrane synthetically, which is very stable.
So the job of the membrane is to separate things.
Yeah, to separate things. So like, and then we put a biological nanopore on this membrane, which it does is basically punch a hole in the membrane, a single hole, very tiny. nanometer level holes. And then we pass the DNA and RNA through this nanopore. So DNA and RNA are both negatively charged.
Yes.
So electrochemistry 111, if you have like a positive charge underneath, then you can pull the negative charge molecule. So that's what we do. So underneath this membrane, we applied positive voltage. So the DNA and RNA goes through this tiny little hole.
So the negative is attracted to the positive and can come through the hole. And if the hole wasn't there, the membrane is impermeable.
Yes.
Right? So we have the hole. Okay.
Yes. That's the force. And then if you don't have the DNA and RNA, so the current is going through this pore the level of the current is what we call open pore current. And then it's equivalent to, like, for example, if you imagine a water flowing through a hose pipe.
Yes.
So water goes through a steady state level. And then when something blocks the hose pipe, and then it blocks the water flow. Similarly, the current passing through this tiny hole or the nanopore blocks when the DNA and RNA is passing through the nanopore.
So that current is carried by ions. Is that
Yes,
right?
correct. Yeah. So based on the composition of the bases A, T, C, G of the DNA or RNA, this current level changes based on the blockage of the current.
So ions are charged atoms.
Yes, if we take normal table salt, as you know, it's made of sodium and chloride. The sodium is positive, the chloride is negative. So under this this setup, the sodium ions would move in one direction, the chloride ions would move in one direction, you detect a current. But because the DNA is also negative, it will move in the same direction as the chloride ions.
But so you've got, okay, I'm a bit confused. You're pulling through, you're basically using electric charge to pull through a strands of DNA or RNA that can be 3.5 billion base pairs long. Yeah. It must take forever.
No, I mean, yeah. So we run this actually quite fast, and we also control the speed using a biological motor on top of this hole. So if we don't have this motor, it will pass through hundreds of thousands bases a second. So currently, then you don't get the high enough resolution, so the accuracy will go down. So we control the movement of the DNA and RNA through this motor. So currently we use about 400 bases a second speed.
Oh, wow. Okay.
And within, so we can run any time that you want to. I mean, until the experiment is done, you can run run this device. So we typically to do the human genome, we kind of advise a 72-hour run, and within that you can get enough information, and it's heavily information rich. So we get this current blockade signal, and then using our AI machine learning tools, we convert this current blockage signal to decode the DNA sequence.
So have I understood this correctly, Lakmal? So basically you're saying that you have a nanopore, which is made of a protein, and then on top of that protein is another protein, which is the motor.
Yes.
And then the DNA comes through. The motor helps to slow it down somewhat.
Correct.
As it goes through the pore, because if it goes through at normal speed, it's so fast that by the time it goes through where the sequence is read, because the differences in the current are quite small between A, T, C, and G, we could get mixed up and we can make mistakes in the readout.
Absolutely.
Is that right?
Yes, correct.
And you said earlier for the COVID virus, you're down to $5 a sample, I think, or £5 a sample. A human genome, how much would that cost these days?
So let's go back 20 years when the first human genome was mapped. Yeah. It cost $3 billion,
yeah.
And it took 10 years, and it was a worldwide consortium. In the last 20 years, even with the decentralized machines, and a lot of the academic research that we leverage came from something called the $1,000 genome. Mm-hmm. And that barrier was breached in about 2012 to 2014. So we went from 3 billion
to 1,000.
And at that point, the technology was outstripping Moore's law.
Yeah, wow.
It was moving faster. So you have this real technology explosion. And today, we can do a fully comprehensive genome for $600, $400 to $600. And that has everything in it. And so that kind of is, people now are looking at rare disease. So these children growing into adults can be on a 5 or 10 year diagnostic odyssey where they're trying battery after battery of tests. You can do a whole human genome and you can find these multiple modes of genetic aberrations that cause the disease in these rare disease people. It doesn't necessarily mean you can solve them. Sometimes there can be some quick fixes.
Yes.
Just a diet change can change quite dramatically. But what we're doing now is building the atlas of rare disease. And as we understand that better, that's something that pharmaceutical companies and drug researchers can really get at. So it's affordable, it's accessible, and it's becoming global.
So it's a fantastic story, and clearly it's already revolutionizing diagnoses. And as you say, you're beginning this journey. It's the bit of technology that we hear about as scientists, but more and more people are going to be discovering this in their everyday lives going forward by the sound of it. So many congratulations on that for the company. What next? Where do you go next? I mean, you've solved it all, haven't you?
You've been out to space. You've been out to hospitals all around the world.
I think, another thing, it's more about this, this country creates phenomenal academic excellence. It creates phenomenal innovation. And I was involved with a company called Medisense that was sold to the Americans in the foothills of commercialization. And we vowed to try and become a British tech, global tech player out of London. So we floated 3 years ago. At that point, I was asked if I was going to retire and head for the hills. And I said, no way, this is just the beginning of what we can do. And the use cases, not just as we've talked a lot about clinical applications, but in environmental, in food safety, in pandemic preparedness, in biosecurity. We're just at the tip of the iceberg. And the opportunities are phenomenal. And I do want to share one with you. And that is today, there are something like 18,000 children with brain tumours. It takes 4 to 6 weeks to diagnose them. All the samples go off to a lab in the southeast of England. Somebody at the University of Nottingham pioneered a workflow where they want to go in and interrogate the tumour. It's routine practice before they decide on treatment.
So you do the biopsy.
So they take a biopsy, and in 2 hours, it gives them a guide on which way to direct their treatment. Do they want to cut it out? Do they want to do chemo? Do they want to do something different? That interoperative tumour profiling using what I talked about earlier, methylation, the C base. So this is characterization of cancer with this chemically corrupted C base to tell you what type of cancer it is. And that's just the start. We see a breakthrough like that every single week. And, people always say, what gets you out of bed? Why are you still doing this after 20 years? Because you go to work every day. The clinicians I met at Guy's and St. Thomas's this morning, they are just so energizing. They're so excited about the disruption and the change it will bring in treatment for them. And seeing that right up front and close and personal is so invigorating.
Also going forward, the platform that we have developed is very modular. So we can easily take this biological nanopore for DNA out and put a new pore in for RNA, or put a new pore in for proteins, or put a new pore in for like metabolomics, without having to redesign the entire system. So as a result, we are now I think the genomics field is advancing to include more multi-omics approaches, which means you get information from DNA, RNA, and then next thing is what actually the DNA, RNA changes do in the body. So then you need to analyze proteins. So we are now working on proteomics so you can hopefully very soon you can read protein molecules through this tiny pores in our system, and that will give another layer of information to add into DNA and RNA.
So do you need to unwind? Do you need to break down the structure of the protein to create the string of amino acids, or can you read the whole protein going through?
We can do, there are a few methods that we are developing at the moment. In one method, we do pass the entire polypeptide through the nanopore, but in one method, we chop the peptide into small pieces and then try to detect these pieces so that we can correctly call the protein identity.
It's a really inspiring story. I mean, and congratulations. And it's also fantastic that, this is, as you say, this is a British tech company and it's going to remain British. There are a lot of scientists out there, young scientists who are starting their careers. What advice, who will come up with some great ideas that, potentially have the opportunity to have major impacts on the world, much as you have done. What advice would you give them, having set up 2 successful companies?
I think I get asked this a lot.
Sorry.
I think you have to be so. I still sometimes look back and think, how did I have that much self-belief? Yes. But somehow you just, in the moment, failure is not an option. It's just not an option. And all we ever said about ourselves was, we're a big company. We're just temporarily small.
So Lakmal, what do you're the chief scientific officer, aren't you? So, why do you get up in the morning?
Well, for me, I think I'm hugely passionate about what we can do, especially in low and middle income countries. And also the potential of this technology is enormous. And as Gordon said, at the beginning of the company, for 5 years, nothing was really working, but we never gave up. We kept going. And then also at the right moment, we changed the technology. So we were very agile. And that got us where we are. And even now, we are a very agile company. So we are at the right time, we do things differently. And there is especially to get into proteomics and metabolomics, there is so much to do. There's a mountain to climb. And that's what, keep me up all night. Because the opportunities and I can see how it changed DNA RNA world, and I could see that how can it change the proteomics world as well. And then together it's kind of very, very powerful. Imagine you go to GP's office and when you talk to the doctor, by that time you have your diagnosis, whether you are infected with the viral or bacteria and what antibiotics you should take. And if you can take it to the next level to analyze proteins, that's huge. And we can do that. We are the only company can do all that in one platform.
That seems like a good enough reason to get out of bed to me.
And you certainly were small as a group of people when I first met you. And honestly, it's just hearing the stories you're telling. And also, I think, just seeing in your eyes the personal, clearly, satisfaction you get from talking to clinicians, for me is, it's made my week, really. It's been really, really nice.
We're very privileged to have you come on and chat to us. So thanks for taking the time to do that. Really appreciate it.